Hi everyone, welcome to my channel. Today we're going to learn about cell membrane and ion channels. Okay, so that we can understand the next video, which is transport across cell membrane. So let's see. Okay, so first of all, to understand plasma membrane or cell membrane, first of all, we should know the composition of this. Okay, how things are arranged and why they are arranged in this way. Second thing, you have to understand this thing that we are talking about cell membrane, which is the covering outside the cell. Okay, but when we talk about cell organelles, they are also covered with the membrane with same structure as cell membrane. Okay, so the function of those membrane is also the same, right? So that's why you have to understand in integrated way that we are talking about function inside the cell as well, right? Okay, so let's see what are the composition of cell membrane. So this is basically made up of three things: fats, protein and carbohydrate. If we see here, the fats are the type of lipids are phospholipids, sphingolipids, glycolipids, free fatty acids, ceramides, triglycerides, cholesterols and cholesterol esters. Okay, so these are the major form. So how you have to answer the question if the question come like cell membrane only or composition and function of cell membrane? So how we have to make an answer which can actually made a significant influence on the examiner. So let's see how we are going about that. Okay, so first of all, you have to draw a cell membrane with the bilayer structure. So you have to start with the explanation that this is cell membrane having bilayer, lipid bilayer structure. Now, what is this lipid bilayer? First of all, you have to make this thing very clearly explained, right? So first thing, phospholipids. If we talk about phospholipids, so this is a glycerol molecule having a phosphate group and the lipid layer. We know that lipids are water insoluble. Okay, so this end is will be hydrophobic and the phosphate end will be hydrophilic, water loving, right? So how these lipid should be arranged? If we talk about plasma membrane, the outside and inside, we know that there is fluid, intracellular fluid and extracellular fluid. So if we have fluid here, so water loving should be on both side outer end, right? So that's why if we see this structure, this phosphate molecule is the head which you are seeing here, outside and inside, and the inner side is basically the lipid loving part, that is hydrophobic. So this is the head, okay, and these are the tail part. So this point should be clear to you that head and tail portion of phospholipids, they are arranged such a way that water loving or hydrophilic end is outside and inside toward extracellular fluid and intracellular fluid, and the lipid loving which is hydrophilic is interior side. Okay, this point is clear. Okay, so this is all about lipid bilayer. As you can see in the structure, this is not only lipid bilayer but a lot of other things are also present. So first of all, we have to explain the lipids in which lipid bilayer is present. And if you can see here, the lipid bilayer is, there is outer leaflet, there is inner leaflet. Why we have written this thing? Because this is really important thing. Okay, when we talk about cell membrane, the cell is playing some roles, specific roles, depending on that where the cell is present. Okay, so that's how the composition of these lipids in lipid bilayer varies. So outer leaflet, the most important lipids are phosphatidylcholine and sphingomyelin. Okay, in inner leaflet there are more of phosphatidyl inositol and ethanolamine. So depending on what function the cell is playing, the composition of outer leaflet lipids and inner leaflet lipids varies. For example, in CNS, what we want is basically formation of myelin sheath, right? So in those cells, the sphingomyelin composition increases. There are some cells where the signal transduction pathway, the IP3 pathway, DAG IP3 pathway, you'll go through in future lectures. Okay, so their phosphatidyl inositol concentration increases in the membrane. So that's how the composition of outer leaflet and inner leaflet have some polarity, have some functions specifically to the location and specifically to the cell function, right? Okay, so here two things you have to make clear. First about lipid bilayer and second thing about the composition in outer leaflet and inner leaflet. Third things are, there are some cholesterol collections here which are called lipid rafts. Okay, these are cholesterol and sphingomyelin collections, some percentage of sphingomyelin also, and these collections act as some cavula, or they can also act as receptors. They can also help in transport mechanism, which we'll be discussing in next video. So this is all about lipids. Now let's talk about proteins. Now, apart from these bi-layer structures, you can see many globular structure which some are crossing the membrane, some are not crossing the membrane. So all these are proteins. Okay, so proteins are of two type: integral proteins, which are the transmembrane proteins, which are covering whole the membrane, right? And there are peripheral proteins which are either present on outside or present only inside, right? So these are basically peripheral proteins. They can be on outer, they can be on inner. So what are functions these proteins are playing? Why some proteins are crossing the membrane, present throughout, why others not? Okay, so there are functions of protein, which is really important question in general physiology, which comes most often. So what is this function of proteins? So let's see. So there are proteins which, the transmembrane proteins I am talking about. So if they are crossing the whole membrane, they act as ion channels. They also act as pumps. They help in active transport or passive transport mechanisms. They act as carrier, for example glucose transport, amino acid transport. They, if the proteins are integral, not integral but only peripheral, then they act as receptors. They can act as receptor for some molecules, or they can act as receptor and help in vesicular transport. Okay, they can act as receptor for some signal transduction pathway. Now signal transduction, when you will read, you will understand that the peripheral proteins not only outside the cell membrane but inside the cell membranes also act as some receptors or some proteins in signal transduction. They can act also as adhesion molecules. What that mean to you? That sometime a cell need to do some functions. It will have to attach to some foreign or some surrounding cells, and now for that attachments it need adhesion molecules. Here gap junctions are also example of that, tight junction are also example of that. For example epithelium in GIT, with the help of tight junctions they all are acting as barrier in GIT, right? So there are some cell recognition molecule for immunity. Actually we have to recognize whether the antigen is coming, that is foreign or our own. Okay, so cell recognition molecule also. Their blood group antigens, we know that on RBC membrane there are some H antigen and then blood group A, B, that is recognized with the help of subsequent attachment to that H antigen, right? So blood group antigen, for that also cell membrane is very important, and the thing membrane proteins are important. Okay, so this is all about functions of plasma, the membrane proteins. Okay, now at last let's see just ion channel also. We are talking about these proteins are acting as ion channel. So ion channels are of four type. First is a ligand gated channel. Ligand gated channel, what that mean is basically one ligand. Ligand is nothing but a substance. A substance, when it bind to the channel open up. So that is ligand gated. Ligand is basically gating the channel. Example is acetylcholine receptor. Okay, phosphorylation gated, the second example. That means if ATP bind to this, ATP comes and the phosphate ion, phosphate molecule bind to it. Once this happen, the channel open up. Okay, so that's why it's phosphorylation gated. The examples are sodium, potassium, ATPase. Okay, then there is voltage gated channel. What that mean? When membrane potential inside become positive, this channel open up. So that is voltage gated channel. Examples are voltage gated calcium channel or voltage gated sodium channels. The last is stretch or pressure gated. What that mean? Whenever mechanical pressure, you will touch skin and the channel open up and cation influx occur. That's why this action potential generated reaches our brain, and we came to know that we have some touch sensation here. Okay, so basically the same thing happen here. Whenever touch or pressure is given to the area, to the channel, there are fibrous tissue which basically stretched. These collagen are stretched and the channel open up. So this is stretch sensitive channel, and the examples are baroreceptors. So baroreceptors, you will see in CVS how basically baroreceptor, they stretch whenever there is high BP, and they are sensing that BP is increased and the respective mechanisms acts into play. Okay, so these are 4 types of ion channel. If, for example, if ion channel comes into your exam, so this way you have to draw one ion channel and you have to give one example of each. Okay, so that's all about. Okay, so this is all about cell membrane and ion channels. So next video will be talking about the transport mechanism across this cell membrane. Thank you so much for watching. Please share if you like my videos.